Our third module is about genes in populations. Some would argue the father of population genetics was Wilhelm Weinberg, a German physician who seemed equally comfortable with mathematics. You might recognize the name in association with the Hardy-Weinberg principle. Mendel actually made the first statement of a population's genetic constitution under a system of mating. Genotypic proportions following inbreeding. The Hardy-Weinberg law is an application of the same principle to crossbreeding populations mating at random. We will discuss it at some length in this module. However, the real beginnings of the discipline might well be attributed to some early plant and animal breeders. In the 1700s, folks like Fairchild in England, Mather in the US, and Kreuter in Germany published on plant breeding, artificial hybridization, backcrosses, and F2s, and Bakewell in England laid the groundwork for many modern breeds of livestock, revealing the value of inbreeding to fix traits. Darwin in 1859 and Mendel in 1865 were influenced by these pioneer breeders. Darwin reflected on the effectiveness of artificial selection and extended the concept to natural selection. Mendel concentrated on the nature of hereditary determination. From these foundations were formed two schools of study: the quantitative or biometric school, led by Galton and Pearson, and those who supported the inbreeding, crossbreeding principles of Mendel, Bateson and Castle. The differences between the groups were largely over the size of effects that were evolutionarily important. The Darwinians felt that small changes over great lengths of time were the essence of evolution. The breeders felt simple changes, allelic forms, could result in huge and immediate differences. Population genetics developed the means by which evolutionary forces could be modeled and described. In the narrow sense, population genetics is a simple extension of Mendelism. It is the study of simply inherited traits and how allele and genotype frequencies vary. As the discipline has evolved, it has taken on broader perspective. We cannot talk about complex traits in natural or artificial populations without introducing quantitative genetics concepts. For our treatment, we will try to keep them separate. The development of population genetic theory rested largely on the shoulders of three giants in the field: R.A. Fisher, Sewell Wright, and J.B.S. Haldane. Ronald Fisher published the first of 28 papers on population genetics in 1918, with a paper that introduced the concept of the analysis of variance, or ANOVA. It was titled "The Correlation Between Relatives on the Supposition of Mendelian Inheritance." Sewell Wright first published on guinea pigs in 1908, but produced generalized theories of inbreeding and crossbreeding, path coefficients, and much more into the 1960s. In 1924, John Haldane worked out the theory of selection by considering a single gene subject to selection and mutation. These pioneers were closely followed by a cadre of brilliant scientists who drove the modern synthesis of genetics, population genetics, and evolutionary theory, including the likes of Julian Huxley, Ernst Mayr, J. Ledyard Stebbins, George Gaylord Simpson, and perhaps most influential, Theodosius Dobzhansky. However, it wasn't until the 1970s, with the discovery of plentiful genetic markers, that the science of population genetics moved from being largely theoretical to emphatically empirical, a trend that continues today. The question that drove naturalists such as Charles Darwin, Alfred Russel Wallace, and Henry Bates in the mid-1800s was, how do species come into existence? Population genetic theory provided the answer. The concept of divergence describes the accumulated genetic differences between populations that lead to isolation and speciation. For plant breeders, the mission is to increase the frequencies of favorable alleles at relevant loci in select populations. Population genetics provides empirical models to predict genetic behavior of organisms. For instance, with known allele frequencies, we can predict what genotypes are present in a population and at what frequencies. We can ask questions such as, are all genotypes equally likely to survive and reproduce? Are mating frequencies independent of genotype? Is the population stratified in some way? For example, by proximity, size, or the timing of natural events? To what extent does mating occur with individuals outside the immediate area? Populations are groups of individuals whose relatedness and population structure are usually unknown. For single genes, we are typically interested in the number and frequency of alleles that exist in a population, and the related genotypic frequencies of individuals. From the latter, we can calculate observed heterozygosity, or the proportion of individuals in a population that have two different alleles at a given locus. Loci that have more than one allele are considered polymorphic. Populations may be described by the proportion of loci that are polymorphic. Of course, this measure is a bit of a moving target and depends on many things, like 1. How you define a locus, 2. Which loci you measure, 3. Size of population sampled, 4. Definition of what constitutes a polymorphic locus, recall earlier discussion about frequency of most common allele, and so on. The Hardy-Weinberg, HW, principle is an elegant little equation that tells us a great deal. For instance, in a two-allele system, given the frequency of one of those alleles, we can predict the frequencies of all three genotypes, the two homozygotes and the heterozygote, in a population. We can use allele frequencies to provide a quantitative measure of variation among populations. We can calculate an expected heterozygosity, HE, for a locus. The model is easily extended to 3 or more alleles by simply adding additional terms, such as described above for each allele and genotypic class. Expected heterozygosity in such a condition is simply the sum of frequencies for all heterozygous classes. You should convince yourself of the Hardy-Weinberg model by working through some of the algebra. It may help to remember that in a two-allele system, with A1 and A2, the frequency of the two alleles may be described as P and Q, or P and 1 minus P. So, the Hardy-Weinberg equilibrium model describes how the frequencies of alleles and genotypes will behave in populations from generation to generation in the absence of several evolutionary forces. Why is it important to understand gene and allele frequencies? As Hartle notes in his Primer of Population Genetics, written in 2000, a very practical application of the Hardy-Weinberg principle is that of DNA typing for forensic purposes. How can forensic scientists be so sure they have identified someone unambiguously based on DNA evidence? Let's just do a little experiment. Take a piece of paper and follow along. Human forensic scientists use genetic markers that have many alleles, often 20 or more, and they use a panel of 13 loci. Let's try something less overwhelming. Let's assume we have 5 loci, each with 5 alleles, all of which occur at the same frequency of 0.2. At any one locus, for any given allele, the probability of being a homozygote is 0.2 squared, or 0.04, which is 4%. The probability of any given heterozygote at any given locus is 2pq, or 2 times 0.2 times 0.2, or 8%. You should convince yourself that there are 10 heterozygous genotypes and 5 homozygous genotypes at any given locus, which should add up to 100%. Now ask what the probability is that 2 individuals would have the same genotype at 5 different loci. If we assume both individuals are heterozygous at all 5 loci, then the answer should be 8% to the 5th power. That is 8% times 8% times 8% done 5 times. The result is 3.27 times 10 to the minus 6, or the two individuals would share the same 5 locus genotype 3.2 times in a million. Rather unlikely. The probability declines even more if you throw in a homozygote or two. With 13 loci, each with 20 or more alleles, the probabilities become vanishingly small. The conditions necessary to meet Hardy-Weinberg equilibrium are a reflection of those evolutionary forces that change allele frequencies: selection, migration, mutation, and drift. Equally important is the issue of random mating among individuals in a population. In the real world, the conditions for Hardy-Weinberg equilibrium seldom exist, but in most natural populations they are generally rather closely estimated. Consequently, Hardy-Weinberg is often found not to be violated. Such is not the case in breeding programs where population sizes can be small, individuals chosen for breeding may represent a subset of relatives, and matings are typically non-random. In fact, the goal of breeding programs is to change allele frequencies, so Hardy-Weinberg should be violated if the program is successful. For most of the remainder of this model, we will be discussing the factors that cause deviation from Hardy-Weinberg equilibrium conditions. Having a clear understanding of these factors is essential to interpreting observed conditions in natural and artificial populations and in prescribing management strategies to meet targets, whether they be used for genetic conservation planning or breeding for disease resistance. We begin by discussing violations of random mating requirements. This is particularly relevant to those seeking to conduct meaningful association genetics tests, for it has been found that population structure can significantly bias findings. Admixture of 2 or more Hardy-Weinberg equilibrium populations with differing allele frequencies produces a mixed population that has a deficiency of heterozygous genotypes relative to the frequency expected with Hardy-Weinberg equilibrium for a single population with the average allele frequencies of the 2 independent populations. Conversely, there will be too many homozygous genotypes. This phenomenon is known as the Walland effect or principle. You can convince yourself by simply creating 2 populations with different allele frequencies for a single locus and calculating Hardy-Weinberg equilibrium values for each, and for a hypothetical population with the average gene frequencies of the 2 populations. As Hartle notes in his text, the Walland effect provides a rather interesting paradox in population genetics. Inbreeding exists in the metapopulation composed of the aggregate of subpopulations, even though each subpopulation itself is undergoing random mating and is in Hardy-Weinberg equilibrium. The reason for the paradox is that the population as a whole is not undergoing random mating. There is remote inbreeding because matings occur only within subpopulations, and because these subpopulations are finite in size, relatively speaking, the level of inbreeding as measured by FST gradually builds up. The smaller and more isolated the subpopulations, the higher the FST value. The result is a hierarchical population structure. FST is simply the correlation of randomly chosen alleles within the same subpopulation relative to that found in the entire population. It is often expressed as the proportion of genetic diversity due to allele frequency differences found among populations. In wide-ranging wind-pollinated conifer species, the fixation index is often quite small, less than 5%, while inbreeding species may sport FST values in excess of 50%. A value of 0 implies identical allele frequencies in subpopulations, while the upper limit of 1 implies subpopulations share no common alleles for that locus. The Walland effect reflects the fact that within finite populations, individuals will share alleles that are identical by descent, called IBD, simply by chance, regardless of whether the individuals are closely related or not. This remote type of inbreeding is distinguished from close inbreeding that might be defined by matings among cousins or sibs, or even selfing. Close inbreeding drives populations toward homozygosity quite rapidly. The more closely related, the faster the process, as is shown in the following slide. Interestingly, inbreeding does not alter allele frequencies in the absence of selection. All bets are off if there is strong selection against an allele, as is the case with deleterious recessive alleles. In that case, the alleles are quickly lost from the population. The inbreeding coefficient F represents a proportional reduction in heterozygosity. The inbreeding coefficient F is not equivalent to FST, which is a measure of genetic divergence between subpopulations. This figure, borrowed from the White et al. Text entitled Forest Genetics, demonstrates the rate at which heterozygosity is lost and homozygosity is gained with successive generations of equivalent inbreeding. For instance, under the straight selfing model, as might be imposed in corn breeding, it takes about 6 generations to reach complete homozygosity at any given locus. For outcrossing tree species, such as those typically found in forest conditions, inbreeding is generally a bad thing. It leads to something called inbreeding depression, which is typically manifest in reduced survival, growth, and adaptation to heterogeneous environments. In the photo shown above, again taken from the White et al. Text, the row of pitiful trees in the middle represent a group of progeny derived from a selfing of one individual, while rows on either side are progeny of outcrossed parents. At age 33, the survival of this Douglas fir self family was only 39% that of the outcrossed offspring, clearly a reflection that this selfed parent carried a number of deleterious or even lethal alleles that were protected in the heterozygous condition but exposed in the homozygotes. A few attempts have been made in conifers to conduct several cycles of inbreeding followed by outcrossing in hopes of exhibiting hybrid vigor, such as that found in corn, but with little, if any, success demonstrated. The genetic load in most trees is simply too great for that to be effective. Conversely, there are a few tree species such as torii and red pines that are naturally found to possess very little genetic variability of any kind. Presumably, they tolerate inbreeding quite well. The interpretation here is that these species survived a very restrictive bottleneck event, which we shall describe shortly. We saw previously that Hardy-Weinberg conditions are met if there are no significant evolutionary forces at play. In the real world, such a static condition would likely never be found, though possibly closely approximated. There are 4 major evolutionary forces that can change allele frequencies in populations: mutation, migration, drift, and selection. We will discuss each of these individually in the following slides. Genetic variation begins with mutation. By definition, a mutation is a heritable change in the DNA sequence of an organism. Mutations may lead to changes in allele frequencies, though it is not a given they will do so. Let's talk about mutations just a bit. Mutations may occur in somatic cells, in which case they are not passed on to the next generation, but do affect new daughter cells, or they occur in germ cells and are passed along to the progeny. Though some somatic mutations are important, leading to some types of cancer for instance, they are of little evolutionary consequence. Mutations of any type are rare. For any given locus, the rate of mutation may be 1 in 1,000 to 1 in 100 million per generation, but with sufficient time and population size, the cumulative effects can be large. Mutations are often found to occur in hotspots in the genome. We know the accumulation of SNPs in loblolly pine vary greatly among genes. Some have virtually no SNPs over the expressed sequence region, while others may have 1 every 50 bases. Many mutations appear to have no effect at all, occurring in intergenic regions or at redundant codon sites, third position, for instance. Most mutations are likely to be deleterious and are quickly lost from the population, or are retained at very low frequencies, usually in the heterozygous condition. However, mutations that confer an advantage in some manner, given luck, can establish in a population, increase in frequency, and slowly spread throughout the population. The movement of alleles among populations of organisms is referred to as gene flow. It is the process by which new mutations that have successfully established in a subpopulation are distributed more widely in the species. For most forest trees, we typically think of gene flow as the movement of pollen or seed across the spatial landscape, though for some species that commonly propagate vegetatively, such as poplar or willow, gene flow may occur by broken branches floating down a stream and embedding in the bank to root and grow. High rates of gene flow among populations hinders differentiation and encourages homogeneity, such is typically the case in most widespread wind-pollinated conifers in the northern hemisphere, though for some species, with few disjunct populations such as foxtail pine, gene flow may be severely hindered. Low migration encourages divergence among populations, a condition that leads to speciation, ultimately. Surprisingly, little migration is required to keep populations from diverging. The absolute number of migrants, MN, per generation need be little more than 2 to keep variation among populations very low. Severely restricted gene flow, however, quickly leads to population divergence resulting from Wallen's effect and something called random genetic drift, our next topic. Genetic drift refers to the changes in gene frequency that occur by chance alone due to sampling in small populations. The figure shown here illustrates how populations of different sizes, 12, 24, or 48 breeding individuals, will tend toward fixation of one allele or another over time. All populations begin with allele frequencies of 0.5 for 2 alternative alleles. If starting allele frequencies are skewed, populations tend to fixation much more rapidly than shown here. Drift has 3 important consequences if allowed to persist without other evolutionary factors like migration or selection. 1. With time, there will be reduced genetic diversity in subpopulations due to allele loss. 2. The average level of heterozygosity will diminish. And 3. Subgroups or subpopulations will become increasingly differentiated from one another. The bottleneck effect is an extreme example of drift, such that the founding population no longer genetically represents the original population. Consider situations where relatively rare endemic populations of some organism, let's use the New Zealand black stork as an example, are caught in cyclonic storm while migrating. Only 10 birds survive. Allele frequencies of some loci will be dramatically affected right off, and all loci will subsequently suffer from inbreeding even if the population rebounds. Another special case of drift is something called the founder effect. Imagine a tropical island, void of coconut palms until 2 coconuts are brought to the island by Polynesians in canoes. Those 2 seed may have as many as 4 alleles at a given locus, but in all probability, they may only have 1 or 2. Some alleles may simply not be present. The bottleneck condition is sometimes referred to as the Sewall-Wright effect because of his treatment of the subject in his writings in the 30s. It may well have been called the Gulick effect. Gulick was an American missionary in the Pacific Islands who studied land snails in different deep valleys that had seemingly random differentiation among races. Vernon Kellogg published a paper in 1907 detailing the Gulick work. Sewall-Wright read the paper in 1910 and later developed the concept in an important 1931 publication. Bottleneck and founder effects have resulted in many interesting stories in human genetics. We conclude our discussion of evolutionary forces with several slides on natural selection. Charles Darwin is widely credited with developing the concept of natural selection, as put forth in his book entitled On the Origin of Species in 1859. This concept was formulated over 20 years following his famous sea voyages around the globe. He was greatly influenced by the work of plant and animal breeders and their successes with artificial selection. He was also influenced by the theory of uniformitarianism in geology, which promoted the idea that simple, weak forces could act continuously over long periods of time to produce radical changes in the Earth's landscape. And of course, he was not alone in conceiving this process. Others with whom he was familiar, most notably Alfred Russel Wallace, were developing their own versions of the concept. The essential elements of the theory of natural selection are noted in this slide. Interestingly, and unlike the other forces which tend to influence differentiation between or among populations in one direction only, selection may act bidirectionally. A central concept in natural selection is that of fitness. It describes the capability of an individual of certain genotype to reproduce, and usually is equal to the proportion of the individual's genes in all the genes of the next generation. If a specific genotype confers an advantage, either in survival or reproductive capacity, it is likely to increase in frequency in the population. Maynard Smith defined fitness in the following way. Fitness is a property, not of an individual, but of a class of individuals. A class would be defined as those individuals with a given genotype. Thus, the phrase expected number of offspring means the average number of progeny produced by a given class, not the number produced by some one individual. Fitness may be discussed in terms of absolute or relative fitness. Absolute fitness, WABS, of a genotype is defined as the ratio between the number of individuals with that genotype after selection to those before selection. It is calculated for a single generation and may be calculated from absolute numbers or from frequencies. When the fitness is larger than 1, the genotype increases in frequency. A ratio smaller than 1 indicates a decrease in frequency. Relative fitness is quantified as the average number of surviving progeny of a particular genotype compared with the average number of surviving progeny of competing genotypes after a single generation. The fitness of the heterozygote relative to that of either homozygote describes the concept of dominance, gene action. Thus, we define gene action as being additive, dominant, or partially dominant, important concepts in that dominance influences how selection sees genotypic effects. This slide gives a nice visual illustration of how gene action is related to fitness. Genotype fitness is defined using two parameters, the selection coefficient, noted here as S, and the degree of dominance, noted here as H. In this illustration, allele 1 is presumed to have a relative fitness of 1, except in the case of overdominance. For the purely additive gene action, the A2A2 homozygote has a selection coefficient of S against it, so its relative fitness is 1 minus S. The heterozygote falls halfway between the homozygotes. In the case of partial dominance, the heterozygote may slide anywhere along that line, depending on H. For complete dominance, there is no selective advantage to being homozygous or heterozygous for allele 1. The overdominant situation is interesting in that the heterozygote is more fit than either homozygote, each of which may have different selection coefficients. This illustration, borrowed from Hardy, shows how quickly a new mutation with a selective advantage, relative fitness of 1 for the homozygous condition of the favorable allele versus 1 minus 0.05 for the other homozygote, will alter allele frequencies in a population given alternative gene action. It is interesting to note that allele frequency nears but does not obtain fixation under the dominant gene action model. Why? Though the recessive allele in such a model has a lower relative fitness, it is not selected against in the heterozygous condition and therefore persists. Alternatively, if the recessive allele has the higher relative fitness, it seems to take forever to get traction and increase in frequency. This is because the frequency of homozygous recessives will stay vanishingly small unless some other force is imposed, such as assortative mating. You may wish to work through this exercise, as provided by White et al., to convince yourself of the concepts of absolute and relative fitness. Selection is often described by its predominant effect on a population. Types of selection commonly discussed today include those illustrated here. Directional selection will move a population in one direction, as the name implies. Diversifying selection drives a population to be bimodal, or perhaps to have several fitness optima. The opposite of diversifying selection is stabilizing selection, which continues to drive a population to a single point with decreasing population variability. The neutral theory of molecular evolution states that the vast majority of evolutionary change at the molecular level is caused by random drift of selectively neutral mutants. The theory was introduced by Mutu Kimura in the late 1960s and early 1970s, and although it was received by some as an argument against Darwin's theory of evolution by natural selection, Kimura maintained, and most evolutionary biologists agree, that the two theories are compatible. The theory does not deny the role of natural selection in determining the course of adaptive evolution, from Kimura, 1983. However, the theory attributes a large role to genetic drift. Indeed, as genomics has provided new tools for simultaneously estimating selection effects of large numbers of SNPs, it appears that most of them behave in a neutral manner. This will be discussed in future modules. As we did in the previous module, we close with a brief discussion of linkage disequilibrium. The relevance of the concept is notable for our future discussions of association genetics. The following slide provides a more illustrative description of what LD looks like. This example provides a numerical example illustrating how linkage disequilibrium, LD, is related to allele and gamete frequencies rather than to linkage per se. Shown on the left are two linked genes, A and B, each with two alleles, capital A and B, shown in uppercase blue, and lowercase a and b, shown in lowercase red. While shown as being linked, we do not show the actual linkage distance because here both tight, small r, or loose, larger R, linkage give the same outcome. On the right are unlinked genes, also labeled as A and B, with upper and lower case alleles as in the linked case. While labeled similarly, the chromosome segments are drawn as solid or stippled brown or green. 3 columns are shown, illustrating different levels of LD: none, higher, or lower. Cell values reflect the observed frequency of each haploid gamete: capital A, capital B, capital A, small b, capital B, and lowercase a, lowercase b. When LD is 0, as we've seen, gamete frequencies are the product of the marginal allele frequencies. For capital A, capital B, this is FA times FB equals 0.7 times 0.6, which equals 0.42. Note that the sum of the gamete frequencies in each column is 1. In reality, of course, the observed gamete frequencies may be very different from those predicted by allele frequencies. In the higher LD column here, we see that one class of gametes, small a, capital B, is missing, and that the gametes capital A, capital B, and small b are both at higher frequencies than one would expect based on allele frequencies by themselves. The capital A, small b gamete has a smaller frequency than predicted by the allele frequencies. How large is linkage disequilibrium in this case? In the lower LD column, the disparity between predicted based on allele frequencies and observed gamete frequencies is not as large, but it is still not the same as in the no LD case. How large is LD in this third case? It is important to keep in mind that LD measures only the difference between observed and predicted gamete frequencies. All 3 situations, as shown here, could arise with linked or unlinked genes, just as discussed on the previous slides. In this exercise, 3 hypothetical conditions exist that reflect no, low, or higher rates of LD. The development of new genomics tools like high-throughput sequencing as well as SNP detection and genotyping has seen parallel development of new tools to fathom how the genetic diversity we are measuring got that way. These many analytical tools open the door for evaluating the selective footprints left on populations. Collectively, these new approaches are known as the relatively new discipline of molecular population genetics. We will dedicate another module to this topic later in the course. Suffice it to say that population geneticists have never had so many tools and data to study. The same can be said for plant and animal breeders. We now can envision dissecting the genetic architecture of virtually any trait in any species of interest, hindered largely by financial concerns alone. In Module 4, we move our discussion to quantitative genetics and the study of complex traits. As should be apparent by now, the line between Mendelian, population, and quantitative genetics is becoming increasingly blurred.